even better. The 2002, and for most people, you have to start with their graduate career to talk about their commitment and their passion for science. But Carrie started in high school when she won second place in what used to be the Westinghouse Science Competition, now the Intel Science Competition. Then worked in science as an undergrad with David Blanchard, went to Caltech, kept working in science with Aaron Schumann, their MD, PhD here, did some excellent work with Sino on NF1 and tuberous sclerosis, and now is Carl Deitorov lab at Stanford, which is going to be talking about the very exciting work that she's going to be doing today. I mean, that is doing right now. Thanks, Carrie, for coming down.
Thank you. Thank you so much for having me. So just bear with me while I figure out what I'm actually doing in terms of moving the slides. So I'm going to be here and hitting the button.
Yeah, you can just hit the button.
All right,
You just
perfect.
click.
So yeah, no, thank you so much for having me. It's really a privilege to be back here where I was trained and to get the chance to talk about some of my work. Very excited. So today I'll be talking about distributed neural dynamics at the intersection of memory and fear. The background here is, like we've been discussing, just sort of a nod to the LA freeway situation, which, having grown up here, I was been quite familiar with, but also was meant to just sort of plant the idea of distributed systems and the kind of organization that it takes to make distributed systems work. Because really, when we're talking about memory and fear, these are processes that are very highly distributed across the brain. Oh. Yes. Okay. So what's the brain's memory lane? Memory is actually processed across anatomically distributed neural populations. Here on the right is just a visual to show just kind of the breadth of brain areas that are recruited just to successfully encode a visual image. These distributed neural populations have to interact to maintain stable memory traces. And what that means is that somehow across this brain, neural populations the activity of these populations has to be organized to maintain continuity of the memory, despite the fact that its underlying content and substrates reorganize over time. They'll have the same core sense of a continuous and stable memory, despite all this evolution of the details of the memory, the nature, and its underlying neural substrates. Additionally, the memory is sort of stably maintained and encoded, even though it's very flexibly and adaptively interacting with more transient states like fear. So in the healthy state, you don't express fear or whatever associated affect there is throughout the entire time the memory is encoded. It's sort of expressed in an adaptive and context-dependent manner or situation-dependent manner. So what I was really interested in the work that I was doing here and hope to begin to address is what are the large-scale network dynamics that we can find that can pull together all of this neural activity and help to support some of these aspects of memory processing, its stability and its flexibility. Okay, so to do this, I look towards the stable, intrinsic, large-scale functional architecture of the brain. So when you look at just sort of the ongoing intrinsic activity of the brain, you find that it's organized into separable large-scale networks. These are correlated, these are sort of bound by their correlation in the low frequency domain, which you can see in the spontaneous fold signal. And prominent among them is the default mode network. So that's shown here on top. And just sort of to illustrate how these networks emerge, you can see a couple of the canonical regions of the default mode network there, the prefrontal cortex and retroscenial cortex. And their bold signal is shown down here, color-coded. The y-axis just being the percent of bold change. And you can see this very tight correlation in the activity of both areas as they move up and down over time. Here below is the entire network you can map out by looking at which areas are brought into this kind of correlated structure. And in particular, when thinking about what are the large-scale dynamics that can help to organize memory, the default mode network pops up because it's implicated in autobiographical memory as well as other internal and sort of subjective processing. And its key memory areas form its nodes. So basically, we can now ask, do these stable, kind of consistent over time interactions between DMN areas contribute to the stable but flexible nature of memory processing? So to answer this question, let's ask a mouse. Mice are a great model system because they also have a homologous default mode network. You can see it in resting state imaging. And in mice, there's a number of tools to directly access the neural dynamics across this network, causally and in real time, and while the animals engaged in free behavior. So for this talk, and for my work in general, I focus on optical approaches, including photometry, centering around photometry. And photometry is where you use as a tool where you use a genetically expressed calcium indicator or calcium-dependent indicator to directly measure the activity of specified neural populations. And because of the sort of genetic drive of it, it's an excellent tool that allows you to bridge network level dynamics to microcircuit and cellular functions, and giving you the flexibility to choose underlying neural populations by their genetics, by their function, and so on. So in the past, I've used photometry to begin to dissect sort of complex encoding across circuits and behaviors. Here on top is some work that I did to look at differential encoding across projection-defined dopaminergic subpopulations of substantia nigra. So very, very easily let us tease apart these mixed populations and look at their role in encoding. I was also able to use it to look at this kind of complex social behavior and pull out single neural underpinnings. So in this behavior where we're looking at pro-social approach and the neural underpinnings of it as this rat approaches this other guy to help it out of a trapped state. So for the work I'm going to talk about here, I wanted to then expand this method to be able to look across different sites in the brain, so more directly tap into distributed neural dynamics. So for our work, I focused on key conserved DMN nodes. They're listed here. It's the prefrontal cortex, medial dorsal thalamus, rich spinal cortex, ventral hippocampus. It's kind of these four areas cartooned there, and a comparison area outside of the network, which was the somatosensory cortex. So we used a virally driven strategy, virus that was expressing our nuclear localized fluorescent calcium indicator, in this case GCAM6M, was injected across all these areas. We restricted expression to excitatory neurons and also to the nuclei of cells. And this was important in helping to increase confidence that we were looking at independent neural populations in each area, right, without cross-contamination by projections and things like that. Okay, so this is what the mouse ends up looking like. Each of the areas with an injection gets a fiber implant on top, which is these little silver circles from the top view and a little head cap. And it can be paired to a light path and a camera system, which then allow us to directly drive and measure underlying neural dynamics simultaneously across all of those locations. And this system in particular, I built out to optimize for simultaneous real-time measurement of all of these areas, but also to allow flexible access to both cortical and subcortical sites with equal weight, right? And that gets around some of the limitations of other strategies. It also is set up to very readily integrate with precise neural manipulation and behavior, and that can be optical, which I will show later, or other methods like electrical or whatnot.
The signal you read out here is an average of the sites.
It's an average of.
Do you get each of these sites?
Yeah, independently, yeah.
Okay.
Yeah, so this is kind of the cross section. You see each fiber independently, and then you can measure. Yeah, and it's camera based. Thank you. All right, and just to showcase what these data kind of look like, looking at very stable multi-hour recordings of the spontaneous neural activity across the DMN. This is just looking at a mouse in its little home cage. I'm recording from these 5 sites. This is a sample trace, and the top 4 traces are from areas within the default mode network. This bottom trace is an area outside the network. And so the entire trace is about 2 hours in length. And so from this kind of data, because it's sort of this long and stable recording of spontaneous activity, it's sufficient to be able to start extracting meaningful network correlations, even in a task-free state. So that's what we've done here. We took this data and looked at the low frequency band, where you tend to see these sort of intrinsic networks emerge, and just looked at pairwise correlations. And what you can see for the default mode network areas here on the right and on the bottom is this very clear kind of increased correlation structure between these areas that is not correlated to the out-of-network area. And so this is data averaged across 8 nights for 2 hours of recording per mouse. This correlation structure is specific to this low frequency, is strongest at quiet rest, and so it's very consistent with the kind of dynamics you see in our imaging. So essentially, this is like a validation of the method, that we're picking up a structure in the neural activity that acts very similar to what we see in DMN and imaging studies.
What how fast a frequency can you measure with this system? Like, can you measure delta waves? You know,
You can measure delta, you can measure theta, which I would love to talk more about our findings and the caveats for. Right now, in the theoretical limitation is 10 Hz because we acquire at 20 Hz. But it's not clear that that's the actual limitation of the indicator. I didn't push too far just out of focus on the slow frequency. I mean, the theta, there's like various issues with theta that would be really cool to address, but it can go higher.
Thank you.
And actually I have some neat coherence data between the slow and delta frequencies if you want to talk later. All right, anyhow, I'm very excited to have people talk about this. You haven't noticed. All right, so we wanted to use some of the strengths of our high resolution temporal acquisition, right? We're basically recording in real time to look at how this intrinsic activity across the DMN emerges over time. And basically this is like a baseline characterization of what this network is doing that will allow us to then ask, you know, how does it change during memory? And what we find is in fact that DMN moves between stable states of correlated neural activity. To do this, we took the same data, 2-hour recordings across 8 mice. We split it up into 100-second windows and applied consensus clustering at the low frequency activity to basically find correlation structures or states that the network reliably returned to over time. So these are the 3 states we found. Oh, I should mention this is work. This analysis was done by John Kochalka, who is a really bright and wonderful, enthusiastic person to work with in the lab, and has a history with deep elbow network analysis in humans. So it's a bit of a carryover from the human approach that is done in the Menon lab, but also it's commonly done elsewhere. Okay, so we find these 3 states. They're actually differentiated both in their within network connectivity, like for example, the difference between these states, and in their relationship to the out of network area. So for example, between state 1 to state 3, the relationship with the out of network area is actually one of the prime differentiators. So these states do turn out to be behaviorally tethered. And when we look at recall of aversive memory, we find that it actually increases the occupancy of a highly correlated network state. So to do this, we used associative learning paradigm in mice. This is contextual fear conditioning, diagrammed up here. So we put the mouse in a novel environment in which it later receives an aversive shock. And the mouse basically learns the context and associates it with aversion or fear. When you put the animal back in the same context at a later time point, even though you're no longer delivering the shock, the animal remembers, this is an aversive place, and expresses a fear response. You can test this either at a recent time point, 24 hours, or a couple weeks later at a remote time point. And what you see is that in the course of this time, the memory itself evolves, and additionally, the freezing behavior of the animal in this context changes the structure. So when the animal is expressing fear, it does so by the active inhibition of all movement except respiration. That's sort of a fear state or a fear behavior called freezing. And I'm just graphing it out here as over time, each of these blue bars is about the freezing, and then the animal breaks and starts exploring context. So it goes back and forth. I mean, you can see the structure of that freezing behavior changes. However, there is this very stable recall of the context across time points anyhow. And what we see is kind of correlated with that. There is a very stable engagement of state 1, which is this highly correlated network state. So that's shown below. On the y-axis is percent occupancy of each of these states over the course of exposure of the mouse into the context. And then on the bottom is sort of a labeling of the state. At quiet rest, all 3 states are used approximately equally, but at both recent and remote recall, you have a significant increase in occupancy of state 1. That's really kind of consistent and stable across both the session and the time So,
How did you determine that it's 3 states and not more or less? Was it kind of clustering kind of?
yeah, it was a clustering and it was based on wherever the RAND index sort of asymptoted, which was around 3. If you use 4 states, you didn't get any more explanatory or any more similarity.
So Carrie, should the 3 states in any condition, should they add up to 100%, or are they not covering all the potentials?
They should be adding up about to 100%. So these are about a third of the time in theory. Although the variability, like the box plot kind of.
Are there other states it could be in too? Or-
You can group it into more states. They end up using a kind of smaller percentage of the time. Which was kind of what the question was of how we determine 3. Once you start adding more, you get very little increased explanatory power for variability over time.
I guess my question is, those 3 states, how much of the time does that account for?
We're sort of forcing it. I think you're kind of asking like how big are the clusters around each state, like how close. So you kind of see, so you have basically like this long series of clusters, right, of correlation structures, right? And some of them are not going to be exactly the same, but they're similar. And we're sort of saying we're forcing that into 3 groups of the most, like, you know, if you pick the 3 most similar groups, right, take everything and group it into 3. And they aren't hugely differentiated. There isn't like a group that's very of states that is so far away from another group that there's nothing that kind of looks like one and kind of looks like the other. Does that make sense? So we are sort of forcing it into 3 states. But what we found is if we add more states and start looking at sort of the gray zones between clusters of states, they're not there for a lot of the time. So it's not totally clear if they're different states or sort of very quick transitional things or what. So essentially, if you want to see what's the most stable, most similar kind of organization of the network, it's sort of flipping between these 3. Okay.
And is there a sequence to one state
Yeah.
transitioning into the other and not the third? Like, is there an order?
Yeah, no, that's a wonderful question. We did look at sort of a more causal analysis. I think I'll save sort of the statistics when I have more of the details at my fingertips, because Joan's doing a lot of this. But we didn't find any clear, you sort of could get a structure, but there wasn't any clear sort of behavioral tethering or like it didn't make sense. You know, so it's not clear at that point if there is really any main, I don't know, order or sequence to it.
Sure.
Yeah, amongst these 3 states. What I'll show you for later work, there are some things that now that we know a little bit more about how this works that we could try to look at as far as, you know, areas getting recruited and that. But so far, no. And not at quiet rest. We did look after fear conditioning, so that's not okay. Okay, so let me go back. Thank you. Okay, so basically the argument I'm trying to make here is that we have this kind of engagement of a highly correlated network state that acts as a stable systems correlate of recall, right? Every time you see a consistent recall event, even though other things are changing about it, you see this neural state or this correlation structure emerge. But the question is, if you have this stability in the dynamics, how does all of this restructuring and sort of more rapid shifting in behavioral state occur, right? So as it turns out, on the background of this stable correlation and structure of the network, you have much more dynamic, of course, changes in the overall activity, sort of equally going up and down across areas. And those dynamics are actually what reflect the fear expression very well. So that's shown here. On the right is the mouse's movement in the context. On the left is the mean low frequency DMN activity. And so it's just like an overlay between the 2. This is just sample data from one mouse. The DMN activity is the black line. It's averaged across all DMN areas now in the low frequency band. And now we're looking at total activity, not correlations anymore. And the blue is just sort of movement of the animal. You can see kind of by eye, there's a relationship already forming. So what we did was try to break the behavior kind of in the standard way into bouts or states. We called freezing states anything under 10, which is pretty low to the bottom, that was sustained for more than 2 seconds. So it's when you see the activity kind of flattening down against floor. We called exploration anything over 20 that was sustained for more than 5 seconds. And then so we split the whole data set into those bouts and looked at the DMN dynamics as the transitions into freezing or into exploring occurred. And so that's what's shown. We see the change in the mean low frequency DMN activity on the y-axis. It's all in fluorescence units, just arbitrary fluorescence units, and time here on the x-axis. At the start of freezing, you see the significant decrease in overall neuroactivity across the DMN that you do not see in the out of network area. And in contrast, when animals kind of break freezing and start exploring, you see this increase in DMN activity. But again, it has sort of a smaller and slower dynamic in the out of network area. So you can look at this across mice and across time points, and you see this very consistent pattern where DMN-wide activity or engagement shows opposing shifts that are locked to transitions in fear expression across time. So for both of these time points, here's recent and here's remote. The bar graph is just saying sort of the peak here on the y-axis, peak change in the mean low frequency DMN activity in units of norm fluorescence, either during freezing or exploring. And you see the greatest change in freezing is consistently to go below to decrease activity. The greatest change in exploring is consistently to increase activity. And you can kind of see how this evolves over time on the right, where again, the y-axis is changed in normal fluorescence, the x-axis is time, and the 0 point here is just the start of the affective state or fear state, either exploring in purple or freezing in green. Okay, so let me just check my. So what we find is that fear is actually reflected in distinct low frequency network dynamics from context memory in the DMN. We see that context recall drives stable occupancy of a highly correlated network state, but fear transitions are more dynamically reflected in the network-wide activity shift. Right, so you have these kind of two dissociable network properties that are tied or tethered to independent but related properties of memory and fear. The next question then is, well, context drives the stability of the network structure. What drives these more dynamic changes in activity? So to answer this, I wanted to look more closely at the interactions between distributed neural populations across the DMN nodes. So how are the different nodes interacting to create these overall neural dynamics or overall levels of neural activity? How does the network get engaged? The setup here is very similar to before, except now in one area per cohort, I'm also co-injecting an opsin, which is a light responsive ion channel. I use B-reaches here, which has a shifted spectra that allows it to work very well with GCamp. And that means that in one area, we are going to be both driving activity and recording sort of local and network responses. Okay. Again, each of these areas gets a fiber optic, and that allows us to couple it to the system so that we can both drive and measure the activity of neurons. But what's important to note is in the area where two viruses are expressed, the opsin here in yellow will be expressed in a subpopulation of neurons. And the GCamp, which is our indicator or measure, which allows us to measure activity, is in a different subpopulation of neurons. There is overlap, but there are separable populations as well. And so what that means is that when we stimulate the yellow neurons, we're able to record both the local microcircuit response and the network-wide dynamics, which turned out to give some interesting information. Okay, and again, we coupled it to a light path. I had to adapt the light path a little bit to be able to do this, and that basically gave us the ability to drive nodes in the network in whatever pattern we wanted. I wanted to look at patterned activity and particularly to focus on theta patterned activity, very loosely, just looking at a frequency of stimulation within the theta band. Because of its, clear role in memory and in organizing distributed memory process. Oh, and so the STIM protocol I used is just repeated trains. So each of these bars is a train of stimulation, either at theta, which was 7 Hertz, or another frequency that's typically used, which is 20. Okay, so the idea here was, can we causally map network recruitment? So these are kind of the areas or the nodes of the network that I'm looking at. Sorry, this should be medial dorsal valve. And we're gonna go through and fill in the question. To validate the method, we first just use single pulses of stimulation. So each of these traces above is an average of responses across multiple trials of stimulation at just 20 milliseconds. And it basically shows like, yes, when you stimulate an area, the local response is excitatory. You can see it in individual traces, and across the group, you get a peak increase in fluorescence. So it's just method validation. But now what it allows us to do is start looking at how pattern activity shapes this network function. And we're focusing just on prefrontal cortex, retrospinal cortex, and hippocampus. Okay, so what we find, one of them is that prefrontal cortical theta optimally recruits the DMN. So just to orient, it's a bit of a complicated slide, but it'll follow the same pattern for the next few. The top traces here are average, or all of these traces are actually average responses across all the trains of stimulation. The stimulation train is shown in yellow for 7 hertz and for 20 hertz. The top trace is the local area being stimulated. So this is prefrontal responses to prefrontal stimulation at either 7 or 20 hertz. The next 3 traces are the DMN areas, and then here's the out-of-network area. And then these are samples from one mouse, and then the group quantification on the right. And so basically you can already see both in the samples and in the quantification that there's theta frequency activity in the prefrontal cortex drives both local and network neural populations. And it's significantly better at doing it than the 20 hertz, which also sort of doesn't have that specificity for the DMN areas as well. This level, it's around the 2 to 3 mark. It's sort of corresponding to this kind of late increase in activity. And it's not super clear at that point because it's so laid out, whether it's just spontaneous background activity or some sort of late response to this event. But it's not sort of the clear recruitment of the area that you see other places. Okay, interestingly, the ventral hippocampal theta stimulation optimally inhibits the DMN. So here you see essentially both in the local and network areas, you have a predominant decrease in activity in response to hippocampal theta stim and the ventral hippocampus anyhow, that doesn't generalize to the somatosensory cortex and is much weaker at 20 hertz. And you can see that here and also in the group. There are some interesting different frequency specific effects in the local response as well. Okay, and now into the retrospinal cortex, you see yet another pattern where here you get pretty strong local inhibition in response to theta and to stimulation at any frequency actually, but you don't have any changes in the network. So even though the retrospinal cortex was very good at following stimulation from in other areas or activity in other areas, it couldn't on its own drive the network. And again, in the group, you see this sort of sitting around the level of just like this kind of noisy, spontaneous background activity.
You know, if the mice are awake or frequently.
Yeah, they're awake and they're kind of hanging out in their cage. They're well habituated to their cage. Sort of interestingly, this doesn't work as well when they're moving around and the network isn't as correlated. But that's sort of anecdotal, so I haven't looked at it too closely. I'm building kind of a closed loop setup to try to understand that better. But they're awake, they're doing quiet rest. You know, they kind of groom and they sit.
And how long is your stimulus?
It's 1.4 seconds for the 7 hertz and half a second for 20.
Have
So
you tried more longer?
yeah, I've tried really long protocols, which I'll show at the end. I tried instead of breaking it up, doing continuous trains. So in the long trains, which I'll show a little bit more towards the end, you start to see different dynamics emerge. Yeah. I was guessing that was the question.
Yes.
Yeah. Okay, so here's the map, at least from this data, of the interaction between DMN nodes that has emerged. You see this map within this sort of unitary coupled network of both sources of network drive, so the prefrontal cortex for increased drive, the ventral hippocampus for decreased, and sinks, areas like the retrospinal cortex that follows and potentially integrates activity from other areas very well. And in that way probably explains its high correlation to the other areas, but on its own cannot drive activity in other areas or throughout the network. And And sort of reassuringly, and also what I think is really interesting, the unique contributions of each of these nodes is actually pretty consistent and reflective of the underlying microcircuitry in each of these areas, which shows quite a bit of its own specialization. So for example, the prefrontal microcircuits are really specialized and adapted to maintaining and propagating excitatory activity, right? There's a lot of excitatory recurrent connectivity. The synapses facilitate, they don't adapt. That's kind of one of a number of works showing that is off on the left. In contrast, so it sort of fits this idea of a very excitatory area that drives the network. The retrosplenial cortex, in contrast, has a highly inhibitory dominant connectivity, with a lot of local connections being between interneurons and the excitatory drive coming in from outside of the area. So again, that sort of fits with what we're seeing and doing functionally in the network. And in fact, even the neurons themselves are well adapted in terms of their physiological properties for reliably following inputs. And then finally in the hippocampus, there's sort of this gradient from dorsal to ventral where the more ventral you go, the same patterns of stimulation will recruit more and more inhibition, or at least decreased activity. So that's something I found quite interesting. And an area that's been amenable to study in more detail with this approach, and I plan to in the future. But for the purposes of sort of looking at the questions of what drives these dynamics during fear recall, which is what we embarked on this mapping for in the first place, it does also give us a few key principles that help explain what's going on during memory, or at least during the fear expression portion of it. Because what we see is within this functionally coupled network, prefrontal cortical theta emerges as a unique driver of network recruitment. And the same stimulation that drives this network engagement doesn't actually change its correlation structure, right? Well, all areas are being engaged together and the overall correlations remain high. So it's sort of a dissociable driver of just the overall activity level without really modifying the state or the correlation structure of the network. So that positions prefrontal cortex very well to regulate the kind of activity shifts we see during transitions in fear state. So of course, we wanted to test this directly. We wanted to test this directly by looking at prefrontal control of DMN-wide excitation and fear state transitions during aversive context recall. Essentially, we're doing a very similar experiment, but instead of the mouse being in its home cage, it's now going to be tested just during the recall portion of fear learning. So when it's being put back in the context. And we're using a more prolonged stimulation pattern at 7 Hertz. So it's basically 1 minute blocks of 7 Hertz stimulation in the off-on pattern. And what we see is that, in fact, medial prefrontal cortex stimulation does drive both the transition away from the neural and behavioral signatures of fear. And so by this, I mean both the behavioral expression freezing gets shifted over to exploring, and then the DMN dynamics themselves are recruited in sort of a time lock manner at the least. So that's shown over here on the left. And this is again for a sample animal. This is during recent recall, so it's sitting in the context. The right is the mean low frequency DMN activity shown here in black. We're pulling out the prefrontal cortex right now to not contaminate with our own stim. So it's just sort of other DMN areas. And then the blue is the activity of the mouse. The yellow bars are the periods of stimulation of prefrontal cortex. And you can already kind of pick out by eye what the effect is, but you can look at it in more typical terms, looking at percent freezing or the percent of time the mouse is engaged in this kind of fear behavior, both during periods of stim and not stim averaged across blocks. And you see the significant decrease in freezing during periods of stim. When you look at it more closely over time, so on top, we're looking at mouse movement, on the bottom at the dynamics of the network itself. This is averaged across the stimulation blocks and for the entire length of time. And so as long as the stim is happening, you see a sustained difference in mouse movement that's increased during stim here in yellow and decreased when there's no stim here in black. Okay, so coupled with that, you also see time lapse to the stimulation, an increase in the activity of the DMN, that's shown here in black, that doesn't really generalize outside it. So the out of network area is flat. Okay, so what this tells us, kind of closest to the data, is that the prefrontal cortex theta stimulation does drive the mouse out of the fear state. It's engaging less in freezing and more in exploring behavior, even though it's still in the aversive context. It also simultaneously increases the low frequency neural activity across the DMN in a time locked manner, meaning as long as the stimulation is there, the activity stays elevated, the network stays recruited. Okay, so, and importantly, this kind of stim pattern doesn't have any effects when the animal is just in its home cage. So all of these effects are really contingent on the animal being engaged in some sort of aversive recall. Okay, so now putting all this together, I would argue that during recall of fear memory, the prefrontal cortex really uniquely drives network-wide activity and associated transitions out of fear state, sort of affective control, that's in the setting of memory. This positions prefrontal cortex to couple fear transitions with network-wide activity during recall of an associated memory. So it's sort of being positioned very well to manage the gating between the memory itself and its ability to trigger a fear response. And I think this has quite a few implications for its role in managing this idea of sort of the flexible expression of associated fear in the setting of, you know, an aversive memory having been encoded.
Can I ask you one more question
Yeah,
on the
of
last slide? So in the initial slides for the states, you showed that the 20 hertz was actually more specifically related to the DMN network, where 7 hertz was activating also the outer nerves. Was opposite, was 7 hertz was the
Yeah, for all of these, 7 hertz has been more specific. You do see activation to 20 hertz. It's smaller, and sort of looks equal across areas.
Okay, and so if you, that's why you should chose here 7.
Yeah.
Just give.
Yeah, that's right. I mean, there was sort of this plan of trying to look at 20 hertz for comparison to differentiate local versus network effects, because that always becomes a problem with these experiments, right? Is what's caused by the local drive on the prefrontal cortex versus its effect on the network. Those are still in planning. This took a little bit of finicking. So after this, I also ran these mice with shorter periods of stimulation. In part to look at sort of hippocampal control and renal effects and all of that, which I have that data if you want to look at it. But these mice went through. But yeah, that is an excellent control. I think one that is pending.
And why 7 versus 20? Is it like roughly like a theta?
So 7 is like smack in the middle of theta.
Yeah.
I didn't want to mess around with like shifts between low and high theta and all that. I also did look, kind of alluding to your question earlier, I did look at theta power or at FFTs across the spectrum for all of this data. And you do see this kind of interesting bump in theta power right around 7 during recent recall that then generalizes to the whole network during remote recall. And it sort of fits very well with this idea of sort of, you know, the hippocampal drive at recent times and the more network-based drive of memory encoding at recent time. So I stuck with that. That's kind of where the 7 comes from. The caveat to that data, which is why I don't show it too much, other than saying it guided this choice, is that it's very hard in theta with photometry to pull out like respiratory artifacts and stuff. They all fall right in that range. So it's kind of, you know, it's difficult to imagine respiratory artifacts explaining that whole pattern, but I can't also say with certainty about what data is doing. So I'm sort of work that part is a work in progress. What's going on with the data? But that shows that was the 7 hertz, and then 20 is just the very typical frequency that the opsin responds to very well. So a lot of papers have used it, so it allows us to compare it to just like other people who've stimmed these areas. Good question. Yeah.
So I might have missed your earlier definition of theta from your earlier experiments.
Just said 7.
And so it's your initial experiments were showing you 7 and then you stimulated it 7.
Yeah, there's a wider band. The peak just happens to be 7. Theta in general, people will go anywhere from like, I don't know, 4 or 5, sometimes in delta and sometimes in theta, and it goes up to around 10. It's sort of a broad range. I just kind of stuck in the middle.
Yeah, it was sort of, you know, thinking more of humans where we define theta more commonly as 4 to 8.
Oh, really?
So-
That's interesting.
So 7 would be on the high end or even low alpha.
Oh, really
for humans.
interesting. Yeah, like in experiments where people look at sort of the shift between low theta to high theta over the course of memory recall, like before and after memory, or differentiating exploring theta versus recall theta. There's this kind of shift. So 7 is right in between the two bands. And some people do go as low as 4, but also that 4, that kind of 4 or 5 frequency, I've seen separated out as delta, and also it's sort of a frequency that does different things, sort of syncing the prefrontal cortex and amygdala in some work during fear expression. And actually in my data too, the recent time point, there's a lot of coherence between the 4-5 frequency and these infraslow dynamics. But when you go out to remote time points, the coherence shifts to theta. And it's the infraslow theta coherence that predicts behavior, like memory recall at the remote. So it seemed like it was functionally a little bit different. Although I think, it's under debate exactly what theta is and all of these
If I
things.
can jump in also,
Yeah.
I think in the relationship of theta in humans to rats versus mice, and at least the Bludaki work
Yeah.
points in the direction of theta because these are smaller brains,
Yeah.
the frequency is slightly faster, so the rhythms are moved up in relation
Oh, that makes
to
sense.
humans.
Oh, yeah.
So I think the 7 is actually probably pretty critical.
That's kind of a general shift. I think the side. Yeah, I mean, there's this kind of idea, right? And at least theoretically pans out that, and in practice matches that the longer the area, that's sort of what tunes the frequency, right? So the shorter circuits are a higher frequency synchrony, and the longer ones, like the DMN, is really long projections that unite some of it. And that's, when I first started looking at this, people were like, well, of course, it's the long projections. That's what theta is. That's what's really tuned to theta. So yeah, that makes a lot of sense. Just the mass. Okay. What was I doing? Oh, okay. So yeah. So this is just sort of starting to get into more of the next direction and making inroads into this idea of understanding essentially affective control, right? In the setting of aversive memories that are sort of already encoded, what is it that allows, or is it prefrontal cortex gating of their ability to, is it the prefrontal cortex gating their ability to really trigger affective state, whether it's appropriately or inappropriately in the setting of pathology? So to look at this, so to look at that question and kind of start to build a foundation from which to address it more directly, I wanted to see how some of our approaches that we use in interventional psychiatry affect the prefrontal cortex. And because in interventional psychiatry, as this group knows very well, we're able to really modulate mood and associated affect with these prolonged trains of stimulation that also target prefrontal cortical subregions. Arguably kind of homologous to the medial prefrontal cortex area that I'm looking at as well. So I wanted to leverage this idea because here is a stimulation pattern that we are targeting to prefrontal cortex that we know modulates affect, in the setting of disorders that have a lot to do with memory and cognition. And so if we have a better understanding of how it affects prefrontal cortex, that might really give us some new insights and inroads into the way this microcircuitry couples memory and fear, or memory and affect. So that's all just sort of to set up this experiment, which I'm showing here. This is data from a mouse now, where I'm using the same paradigm or a similar paradigm to the typical 10 Hz paradigm that's used in depression. And delivering it to the medial prefrontal cortex. These are local responses that are being recorded. So these are 10 Hz trains. Each one is shown in the gray bar throughout the period of the gray bar, about 4 seconds worth. And repeated 75 times. These are long protocols, like 3,000 pulses total. And what we see is that over the length of the protocol, you start recruiting this much more complex circuitry. So initially, in response to the first, and just to mention, this red is kind of an artifact channel, so you can ignore that. It's a little bit more difficult to subtract out, so I'm still working on those methods when there's so much stimulation together. But the blue is what we're interested in, and that's sort of the fluorescence measure of the neurons themselves, the neural activity itself. And so what we see is early on in response to the first train, there's essentially a monotonic recruitment of excitation. Every stim pulse gets recruits more excitatory response from prefrontal cortex. But as you proceed, even early on, but certainly towards the end, you get this initial bump followed by a really long decrease in activity of prefrontal cortex that covers the brain. And then there's this kind of escape and an increase in activity that shoots up until the train ends. And so this is really suggestive of plasticity that recruits kind of different microcircuit elements. And so thinking back to kind of the slide where I was talking about the specialization within the prefrontal cortex, this is a good foundation to start to look at, well, are we recruiting more inhibitory control? Are we recruiting different subnetworks that are more or less adapted for maintaining excitatory activity, and so on. You can look at different layers, different connectivity, and so on. So this is kind of the beginning of some inroads towards starting to answer that larger question of what is the local shifts in prefrontal function locally and its ability to tune the network or couple with other network targets. We do additionally find that the same protocols that cause this kind of shift locally do increase the tuning of the ability of the prefrontal cortex to drive the rest of the network. So over the course of this protocol, prefrontal cortex recruits the network and drives it more strongly, and also has more inhibition or leads to decreased activity in the out-of-network areas. Creates more of an anti-correlation. Looks a little bit more, if you think to the sort of state 3 kind of network state that was shown early on, where the network itself was very correlated, but kind of anti-correlated with the out-of-network area. So that's the data shown here. Here we're just showing 60 trials of stimulation. The horizontal sort of a heat map where each row tells you the strength of response of each of these areas to the stimulation in the prefrontal cortex. And so the trials are sort of stacked by rows. The first trial's up here, the last trial's on the bottom. And so you can see this progression over time in somatosensory cortex, prefrontal stem increasingly recruits inhibition or is less able to recruit excitation. We can't really distinguish, but you get much more of depression of somatosensory activity. And in the rest of the network, you kind of consistently get an increased ability to drive the network. Okay, so, together, both of these slides are sort of me building an approach to understand and target prefrontal microcircuit components and large-scale coupling in a way that may give us some inroads into into the regulation of affective state.
Question.
Yeah.
Is it a particular subregion within MPFC that you're targeting, or?
Yeah, that's a very good question. There's a particular layer, it has W2-3. I did a full cohort in the deeper layer, and you get very nice local response, but you don't get any network drive. And that's kind of consistent with some of the connectome-based sort of Allen Alice kind of stuff, where they say the DMN sort of connects with DMN areas connect with each other more in the 2-3 layer.
But do you think you're like, aside from layers, like, dorsal?
Dorsal, definitely dorsal. So I tend to err on the side of dorsal, so that if I do get leak, when I do get leak over, it's into the anterior cingulate. So it's sort of anterior cingulate prelimbic targeting in general. I do think it works less in infralimbic. There's quite a bit of variability. So a lot of these effects in prefrontal cortex, especially, it seems like you have to hit the right sort of subnetwork of neurons. And it's not fully explained. It is explained by how far out of that region of anterior cingulate to prelimbic. Like if you're out of that region or out of that layer, you don't get these effects as strongly, even though you see local recruitment. But there's also something else, like there's variability with respect to some version of the subnetwork you're recruiting. And that's kind of why I'm very interested in trying to find different ways to access which components and like sort of what are the physiological properties of the neurons that are being recruited by the stem, because it does seem to matter as far as their ability to drive. My answer. Yeah. Okay, so basically this is the model that the data supports right now. Low frequency neural dynamics across this intrinsic large scale network, the DMN, sit at the intersection of memory and fear. To be very simplistic about it. We see dissociable regulation of stable correlation structures by context, which are coupled to, and that's kind of a cartoon here, where context can reliably, stably, and consistently over time drive this correlation structure. And they're coupled to control fear responses by these rapid sort of prefrontal theta driven activity shifts. And so this sets up a mechanism where you can kind of account for both the stability or support the stability of memory processing over time, but also allow it some flexibility to adaptively express or trigger associated fear. Okay, so this is sort of a foundation, I think, both in terms of the method and conceptually, from which we can start to bridge more kind of cellular network and clinical function at the intersection of memory and affect. And that's kind of where, at least I'm very excited about moving things forward, and where the method itself is very accessible to, the areas of the method itself is very accessible and sort of designed to address. So, you know, I've been talking a lot about this kind of gating and interaction between memory and fear. I think fundamentally the idea of how large scale circuits balance these two things, stability versus flexibility, is kind of a general question across, you know, multiple domains of neuroscience. But here's one that I think is important, both for the basic mechanism, but also because it has a pretty clear application in terms of psychiatric disease. And just that idea that affective control or even behavioral control in the setting of an affect becomes such an important target in the clinical setting, such that, you know, even in patients where you do have active PTSD or you do have active depression, if you can improve affective control, if you can improve behavioral control, or have a better understanding of how, you know, those aspects of their function are regulated by memory or cognition, you know, then you can make quite a lot of leeway of progress in terms of that patient's ability to function. And also just by time for other treatments to work. So this is one area where I think it's really interesting and helpful to use clinical neuromodulation approaches, especially as in, you know, a place where you have a lot of patients and you collect a lot of data, and you can start to get a real understanding of how these approaches affect, you know, mood, affect in different disorders, not just depression, but PTSD, for example, where this connection between memory and affect is quite the center of the disorder almost. So I think in this kind of setting, you can collect a lot of this data and get a really good idea of different neuromodulation, the effects of different neuromodulatory paradigms or approaches. And then you can take that into mice to really understand the microcircuit and large scale mechanisms that are being shifted around. And then, you know, look across different paradigms to see exactly how they are, if those microcircuit mechanisms or large scale mechanisms are conferring adaptability to that interaction, right? To the triggering of fear, for example. The other major platform here, you know, relates more to the idea of stability of memory over time, right? So what are the mechanisms that maintain stability over memory over time? I sort of showed here that the correlation structure and the large scale correlation across this network is sort of the key feature, but we don't really know what it's doing as far as how it interacts with the real molecular and cellular correlates of memory. So there's this huge and very rich body of literature that looks at, you know, the molecular signatures of memory, the cellular ensembles that actually contain the memory trace that are distributed across locations, the same locations that we find to be correlated in their sort of large scale dynamics here. But we don't really understand how are these large scale slow dynamics shaping their recruitment. So for example, when the network is correlated, does it make it easier to pull up the memory trace? Does it protect those cells that encode the memory from being recruited by other things? You know, so you can start to look at those interactions more directly. And especially in a place where a lot of the data and methods that really target both the molecular control of memory and these cellular ensembles were developed here. I think it's very well, it's kind of an easy step then to integrate this kind of approach and start to look at the interactions between large scale dynamics and more cellular and molecular processes. And the method itself it's an easy step to do that. You can implement more functional methods to sort of capture just neurons that are active during memory recall or during memory training. You can look at how those neurons interact in one area with the overall network dynamics across different areas. So you can start to get more and more specific about which neuropopulations you're looking at distributed across the brain. You can do things like modified excitability of specific neuropopulations, say prefrontal cortex, hippocampus, or other areas, and use even a CRED-based system, which has been shown to really attract memory to those cells and see how does that affect long-term stability of the network or overall network dynamics. And this way you can really start to provide a better understanding of what it means for this network to be correlated as far as what it's doing to organize the activity of underlying sort of neural substrates, underlying cell populations containing the memory trace. Okay. So that's kind of the second area I think is very amenable and very easy to do, especially in a place like UCLA. And I think more broadly, this is a system that I'd like to develop into a more translational pipeline that very well set up for integrating genetic to systems mechanisms of cognitive disorders with preclinical and clinical understanding. We have a range of cognitive disorders. I worked in neurodevelopmental disorders in my graduate training. There's of course cognitive decline at the other end of life, and associated with different disorders as well. And I think, you know, you can broaden very easily with these approaches to other behavioral paradigms and to other mechanisms that are implicated in cognition, you know, beyond the DMN. So in the past, I've looked at cholinergic control of memory consolidation. We can start to look at how, and you know, and I focus specifically on the medial septal system, which is one that deteriorates very early on in the process of Alzheimer's. So we can start to look at how that system, which I found did control memory consolidation in a kind of a bidirectional way. How does that interact with large scale network properties? How can that be? How can you drive the network to sort of counteract the effects of loss of that activity and so on? Okay, so with that, I think I just will give thanks to everyone here for your attention and listening, and for having me. And huge thanks to Carl and to the lab. It's a wonderful group of people to work with and a very exciting place to train in the last several years. This Carl in the back was like some holiday party or something. It's a big lab. Within the lab, John Pachalka and Anish are both really wonderful collaborators, very bright, thoughtful, a lot of experience both within the DMN and translating back and forth between human and animal approaches, and also to carry this out. I work quite closely with Amit Etkin and now Corey Keller, who's faculty at Stanford, the sort of neuromodulation and understanding neuromodulatory mechanisms using human studies. Okay. Oh, and I have some funding, which I'm very, very grateful for. That's all. Thank you. Take some questions.
Yeah. So I have two actually. One, this morning I was listening to a talk from Dean Juan was, you know, pointing out that in just isolated cortical tissue in a mouse model, that he was able to get sort of learning so that into a regular pattern that the circuit started responding. And I was wondering if that could be going on in your optogen, you know, in your stimulation. protocol.
Yeah, like am I in training?
You're basically in training. I mean, that's what his data was suggesting that. I didn't use in training. I would though. I like
Yeah. No, it does look like it is an entrainment phenomenon. I don't always get that. I don't get it often enough that it shows up in group analyses.
Okay.
But I do see it. So again, I think a lot of the variability in this kind of data is from exactly which subpopulation of neurons you're getting, or which subnetwork of neurons you're getting. I don't know if it's based on the connectivity or synaptic properties or cellular properties, but that's something I'm really interested in looking at actually is kind of, you know, how you can relate the ability of an area or a cell of a target population so if you have a target population which you're expressing your opsin and you look at how well it drives the network, if you then do the slicing and look at the properties of that population, can you start differentiating which cell types or which properties are driving the network better or worse? Because it's not that it's variable, it's that some target areas, loosely defined, I don't know what's special about that target, really, really drive the network, like many times more than other areas. and some have relatively small responses. And some entrain and some don't. So I think it's a really good time to transition from sort of looking at all cortical areas as equivalent black boxes and starting to really dive into what's the microcircuitry, and what are the things you want to optimize targeting in order to recruit sort of a large scale network. And what does that do, as far as behavioral or, resilience of that memory trace or function of that network to disruption.
So
Sorry.
second,
Long-
no, I know, but, so when you showed convincingly that you can disrupt the expression of the fear conditioning, but you actually drive it to extinction. Or, so a regular
Yeah.
pattern, would you actually block the expression? It usually decays over a course of like a week or something. And, I was just wondering if you could.
I mean, like
accelerate.
if you pair this with extinction or something.
Well, I'm just thinking if you're just continually disrupting the expression of the fear conditioning.
That's
rather
a
than
really good,
move it
yeah.
to essentially treatment of the fear.
Yeah, no, that's a really good question. So I'll tell you about the data and kind of the way I think about it, which could or cannot be correct. I didn't do the extinction experiment you're talking about, but I did look at generalization. It's a little bit messy because I was comparing the prefrontal stim group to a no stim group. It wasn't like a TE or control or anything like that. And the time points were sort of weeks, but not exactly the same. So that's sort of the caveat. But I did see much more generalization. Much less generalization, so the animal had much less freezing in a context C, sort of a new context that was not the one in which that it was afraid of and learned from shock. So it did a much better job of knowing now I'm in a new place, I don't need to freeze. So yeah, that's definitely suggests that you're right, but actually if you do this in an extinction environment. The idea I had, because you start to get into this messy area with where are you disrupting the memory or are you disrupting the expression of it, right? And I think that's
exactly what I was thinking.
Yeah, and I think, there's in this data, because the freezing kind of bounces back very well. But when you start going to these long-term changes that don't bounce back, it's harder to tell. And I think that's why another reason why it's very important to start to understand like how these dynamics are interacting with, say, the engram, right? So say more something like the engram neurons.
Also some Zoom questions, and
Oh,
we can alternate between local questions and the Zoom ones.
Yeah, oh, just as one last thing. I do think the idea of like, if you're just increasing exploring on its own and you're doing nothing else, what's the point, right? Because the animal's still there and the memory is still there. But even if you can do that, there's been a good amount of literature coming up that the behavior itself can predict the amount of resilience to future stressors and things like that in sort of a stress version of this. And also in theory, if the animal can just do nothing else but explore more, it's already getting more input from the context to better define or encode this context for something else. So if it's in a new context, the more it's exploring, the more it's engaging with that context instead of this internally driven sort of memory state, the better the animal is probably going to be able to sort of have flexibility and shift out of fear because it has more information to push it in that direction, right? So I think like starting to understand sort of these fine steps that come in between the memory and the final expression of, say, fear and how that perpetuates the disorder, because in this case we're talking about PTSD, or perpetuates a healthy resilient state. I think that's going to start to become very important, and that's kind of what I was trying to get at in my little spiel of like what we can do well here, or what I could do well here, and would be very excited to do. Should I have the privilege? Okay.
There were a couple questions. Maybe let's just do this one.
Is that the first question that you found pretty well?
Yeah.
You found pretty like.
Mine's freeze for less time in about yeah. So
Can you read the question?
do mice freeze for less time per bout at remote memory? The rise in DMN low frequency activity at the end of the 10 seconds of remote memory looks like they are terminating the freezing faster. Yeah, the answer is it's a good question. It's a good observation. Yes, that's kind of typical in these casts that in the recent time period, the recall, and that's kind of what I was trying to show with those little bars. But essentially, early on in recall, the memory still, human sort of version is more episodic. The mouse is sort of, you get these very solid, persistent blocks of freezing without as much switching out into exploring. And then over time, you get much more of a switch back and forth. So the overall freezing stays high, but the structure of it changes, so the freezing switches more quickly. And yes, the DMN dynamics follow that very well. There is this definite piece of this where the longer the network stays up, the longer the freezing persists. There is like an integrator function in the way the prefrontal cortex drives these dynamics and how they relate to the behavior itself. And then in some, in some of the other data I've collected looking at STEM of say hippocampus or spinal cortex, you see that is also unique to the prefrontal cortex, its ability to sort of integrate over time in order to drive these behaviors, if that makes sense, to both recruit the network and drive. So thank you. How do you know they're awake? They look awake. I mean, that's a good question. The idea, I don't know about it because the idea of quiet rest versus sort of passive immobility versus active freezing, like sort of in the behavioral world, being able to distinguish these things that look pretty similar is a point of discussion. These aren't, when I did the early sort of baseline recordings when they were in their cage, they had been well habituated to the whole system. I was doing 2 hours a day for multiple days before I did the actual recording. The resting state dynamics were very stable, but yeah, probably some of those quiet rest periods were asleep in the sort of baseline dynamics that I looked at. But by the time I got to the later data, I just didn't do long enough recordings. The mice never really looked as like there wasn't a ton of quiet rest, and in the context itself, there wasn't really many periods of quiet rest. Yeah. That's probably my best answer. It's not a very good one. Okay, let's see. When you drive 7 hertz in the OFC, do they explore more? One might expect that since high, low frequency DMN activity happens during exploration. Yes. Oh, thanks for answering my question. Yeah, no, it's a really nice connection that actually, theta is associated with exploring, right? And it's nice that here where the prefrontal cortex is driving exploring and increasing these dynamics, you would I haven't recorded it, but you might expect that at the same time you have increased theta, which is sort of the driver of these dynamics. So it sort of makes that last piece causal, although I have to include in, the finding from other people. But yeah, I'm not quite saying that. Okay, I'll just leave it at that. Okay, I'm not clear on how trains 70 to 75 differ from trains 5 to 6. The pattern is the same in that early on you do start seeing more complex dynamics recruited. I think this is for the TMS STEM data that I showed at the very end. So it is pretty quick that you start recruiting other elements. It's really just the first train or maybe 1 or 2 trains, depending on the animal, that shows kind of monotonic recruitment. And then you do start bringing in other elements. It's just that the longer this protocol continues, the more trains you give, the stronger the decreases are and the stronger the increases are.
I have a question. Just to be clear, the experiments at the end you did with Corey, was that TMS or was that optical?
It's optical, yeah. So all of the stimulation I'm using is
TMS.
optical, but it's the same paradigm, yeah.
So I'm curious about the potential of that. I don't know the technology all that well. I don't know how densely one can pack these kind of probes on the cortex of a little bitty brain. But you were talking about mapping. And of course, that's one of the hot topics in human work these days is what's the best critical target to hit. So can you, in terms of the density of these probes that you can place, what's the technical capability of placing a number of these on the cortex? And do you have the capacity to stimulate and record from each one?
Yeah, I was actually talking with David about this over lunch. They have these kind of fiber arrays, kind of mimicking the way LFPs used to have or still have, you know, wire, is that the right one? Oh, or mine. The way LFPs used to have arrays, so you could record from different areas and have sort of a matrix, they're sort of doing the same thing with fiber. So you have these more densely packed arrays. We were talking about bundles, but I also was just reading a paper, although I can't remember the author on the spot, but where it was really sort of an array of fibers closely packed to try to get multiple within a cortical subregion, and sort of at different, they had them diagonal so you could get different depths and things like that, and you could custom fit them and stuff like that. So you really could, they're still all independent, so you get a sort of independent measure from each of the fibers, but it's just mechanical the way you're bundling and implanting them and coupling them and all that. It's a mechanical problem, really. And they showed recordings that look pretty nice from that. So it is doable. You can use things like galvan mirrors to then direct your light just to single fibers within that. You're still probably not at, I mean, you're not at the level of single cell. But yeah, it's doable.
Any other questions? Looks like I give you a break now. Great talk.
Yeah, thank you.
It's great, I really enjoyed it.
Oh, thank you.
I've got to learn to something else, but I'll see you.
Yeah, absolutely. Thank you so much.